We will continue our discussion on cellular energy metabolism. We ended our previous video with this slide where we were discussing the overarching goal of any weight loss plan is to get someone into a healthy body composition. We want to burn excessive fat so that we don't have too much fat in the body for all of that long list that we came up with of why obesity is a challenge to the physiology of the body. While at the same time, we want to save as much protein as we can. So before we go any further, let's just ask a very simple question. Can you just eat less? We're eating all these calories, we're eating all of these macronutrients that we're eventually going to store. If we just eat less, would that do it? If we could get ourselves to be successful in the starvation diet, would we lose weight? And I love this study. It's an old study, but it gathered data from 6 obese males who were put on a starvation diet. So what we're looking at in this figure on the bottom is the caloric intake. And you can see they recorded 8 days before they started the starvation diet of baseline measurements. During those 8 days, they recorded the average oxygen uptake. This middle panel really helps us to get our mind around what their metabolic rate was. On average, how much oxygen were they using to make ATP in their body? And then they also measured their body mass in kilograms. On day 8, they began a starvation diet. They switched to a 450 kilocal diet. And they recorded what happened in the subsequent days that followed. When they ingested fewer total kilocalories, their body weight did go down. You'll see there's 2 lines on this graph. The black dots with the black line is their actual weight loss. And the green dotted lines are what would be expected if we just purely did the math on how many kilocalories they were taking in. But you'll see that as the diet progressed, not only did their weight go down, but there was a progressive decline in metabolism as well. In fact, the progressive decline in metabolism was greater than the decrease in body weight. So this is that difference we see in the top panel between their actual weight loss and what they expected based on the numbers. They didn't lose as much weight as expected because the body reacted to the starvation diet by lowering its metabolic demand. If you have a lower metabolic demand at rest, you aren't burning as many kilocalories. So when you look at the total weight loss, if you follow from where they started to where they ended, how much was the total weight loss of this program? You can see that more than half of the weight loss occurred over just the first 8 days of the program. And after that, it just completely plateaus out. When the body perceives that it is starving, it is going to try to save the animal, right? The whole being. So it's going to adjust by lowering metabolism to require less kilocalories when it's not eating enough. So people can see success in those first couple of days of a diet, and then it just fails to continue to work because of this drop in metabolism that makes caloric restriction alone ineffective. So can this patient just eat less? It is ineffective as a dieting strategy. Research shows that if you want to lose weight, you need a combination of calorie restriction and physical activity. So calorie restriction would be handled by different disciplines, although we could certainly talk to our patients about the benefit of including that and refer them on. We are the physical activity specialists. Let's talk more about how adding physical activity can aid in weight loss. So we discussed our macronutrients that are stored potential energy in our body to be used for mechanical work. But how does exercise burn those macronutrients? How do we selectively lose stored fat while maintaining lean tissue? You guys have seen this gear shift picture over and over again this semester, and we've kind of always just assumed that if you get oxygen to the mitochondria, you get ATP. Let's talk specifically, working backwards, on what these processes are so we know how to stimulate them and target them. So let's first start with this energy currency of the cell, ATP. So ATP stands for adenosine triphosphate, which is a universal energy donor. So this energy from macronutrients, fat, protein, carbohydrates, is not directly used for biologic work. The energy is harvested and funneled through energy-rich nucleotides, ATP. And we can see the structure of ATP on the slide here. You have adenine with a ribose, that's the adenosine. And then there are these 3 phosphate groups. And when we talk about ATP being a universal energy donor, it is the potential energy within the phosphate bonds that powers all of the cell's energy requiring processes. There are other high energy compounds in cell, but ATP is by far the most important. So we will almost exclusively focus our attention on ATP. So in this cartoon version, I used little lightning bolts to represent the high energy bonds. And it's the bonds of the outer 2 phosphates. And they're high energy bonds because they release useful energy during hydrolysis. So the release of energy is a hydrolysis reaction. We could actually write the same thing if we wrote out ATP, and if you add water to it, that's a hydrolysis reaction. You can release some of that energy. This hydrolysis reaction requires an enzyme, which is adenosine triphosphatase. I love the name of enzymes. I think they sound so cool. I kind of wish I had named my children after some enzymes, also known as an ATPase. They would love that. That's a hard life, going through life named after an enzyme. So I named them Alice, Emily, and Tess instead. Okay, so when adenosine triphosphatase is available, or ATPase, it can add water to an ATP, and it breaks off one of the phosphates. It liberates one of the phosphates, which frees that energy to be made available to metabolic work. So the ATP, which is adenosine triphosphate, 3 phosphates, becomes ADP, or 2 phosphates, plus that freed inorganic phosphate floating around. And the process of this hydrolysis reaction liberates energy. Energy is free. Specifically, you get 7.3 kilocalories per mole of ATP when you go from ATP to ADP. There is reactions that can take 2 phosphates off at once that liberate just a little bit more energy, but typically what we see in cellular energy metabolism is the breaking of 1 phosphate off, which releases 7.3 kilocalories per mole of ATP. Now, this energy that you've liberated from this phosphate bond transfers directly to the energy requiring molecules of the skeletal muscle pump. So our macronutrients are not used directly for biologic work, but rather are harvested through this high energy compound because we can easily release that energy right when we need it and right where we need it to power our mechanical work. So where does this ATP come from? Well, there is no storage of ATP in the body. ATP is incredibly heavy, so it would be like 75% of your body weight if you stored enough ATP for the energy of a single day. So we would just be like blobs carrying around ATP and doing nothing else because it's so heavy. So to overcome this limitation, cells store a very small quantity of ATP. As you can see in your notes, about 80 to 100 grams of ATP. And to put that in context, the amount of ATP stored would fuel only several seconds of explosive all-out exercise. So instead of storing it, because it's heavy and hard to store, we are very good at matching ATP supply with ATP use. ATP resynthesis proceeds uninterrupted in the cell. Our cells have multiple pathways to make sure that when we need it, we can make it. So with that in mind, if we could get a little probe to go down into an individual cell and measured their ATP levels, and this is given as a percent of resting levels. So how much of the total amount that we started with do we have? And we measured what happened to ATP during an all-out sprint. You would see a very slight dip in the beginning, and then ATP levels are completely maintained by multiple different pathways. Because if we can't maintain ATP levels, the cell dies. That is the whole processes that we talked about in pathophys that frequently cause cell death and disease. We cannot have ATP levels bottoming out every time we exercise, or we would have massive cell death, because ATP is required for all of the cell's processes and functioning to stay alive. So our body ensures that it can always make enough ATP for what the cell needs. And if anything in physiology is so important and critical that it's the difference between life and death, we're going to have multiple ways to get the job done, which is exactly true in the story of ATP production. So ATP is that universal energy donor that we need to keep a constant supply of because it's too heavy to store, and it's absolutely critical for life. So what do we have? We have mitochondria in the cell. Mitochondria are great at making tons of ATP because they can use oxygen. So the mitochondria is our aerobic pathways, the pathways that take oxygen and convert it to ATP. We will specifically go through the citric acid cycle and then the electron transport chain. But there's more than just that, because the mitochondria requires oxygen. We need to be able to make sure that ATP production goes uninterrupted. And we have talked about times when demand can go up very, very rapidly. We have to have some other way to get ATP. Out in the cytosol, independent of mitochondria, we have enzymes of glycolysis. And these are anaerobic enzymes. They do not require oxygen. We also have our phosphagen system, which can get very quick little pieces of energy off in a one-step reaction. So we have the ability to make continuous ATP supply because we have different metabolic pathways, some requiring oxygen and some being anaerobic. The three main systems that we will go through in detail is the phosphagen system, anaerobic glycolysis, and the aerobic mechanisms. Now, we have multiple pathways to allow for continuous uninterrupted supply of ATP, but they also have their own strengths and weaknesses. So we have pathways that can make extremely high production of ATP, tons and tons of bang for your buck, but they're slow and they take longer. We have other pathways that are incredibly rapid and bada bing, bada boom, almost 1 step, 10 steps, we can get ATP, but the amount of ATP that comes out of those pathways isn't that large. So we're going to burn through that pretty quickly. Daily ATP, because we have this variety of demands across the spectrum, is going to be some kind of combination of both of these. The top of your next page of your notes, we start talking about the energy systems. And the first one we'll talk about is the phosphagen system. The phosphagen system, as it says in your notes, is the simplest and fastest energy transfer. This phosphagen system really helps to overcome the storage limitation of ATP because phosphocreatine is another intracellular high energy phosphate compound. But our cells are able to store about 4 to 6 times more phosphocreatine than ATP. So we're much better at storing phosphocreatine, and we can get ATP out in a simple one-step reaction. So you can see on the slide that phosphocreatine is an enzyme-dependent reaction. It requires creatine kinase. But note that it is one single enzyme, a single one-step reaction. There is nowhere on this slide where you see oxygen. The phosphagen system is anaerobic. No oxygen is required. I think this part is really cool. So it's basically the same as what's above, but I like to see it. I like to write as I talk, in case you guys haven't learned yet. All right, so phosphocreatine needs ADP for creatine kinase to get ATP out. So what is so cool about this system is that if there is a transient increase in ADP in a muscle, it is going to shift the creatine kinase reaction towards PCR hydrolysis and ATP production. So as soon as ADP starts to, even the tiniest little bit, accumulate in the skeletal muscle, it is going to stimulate creatine kinase to create creatinine and ATP. That's the energy. What's so cool about that? Why do I geek out about stuff like that? If you go back to the ATP hydrolysis reaction, when you're using ATP for metabolic work, what do you end up producing? ADP. So we have enough ATP stored in our cells for just a few seconds of explosive activity. But the second you start using ATP for energy, and you start ripping off that phosphate to release the energy for metabolic or for mechanical work, you create ADP. As soon as ADP starts accumulating in the cell, it is going to activate creatine kinase to use the phosphagen system to make more ATP. It is this little self-regulating wheel that just makes sure we always have ATP. That is incredibly cool. So it's very, very fast, but it's also very limited. It reaches maximum energy yield in about 10 seconds. And phosphocreatine is stored right where we need it the most. You will find phosphocreatine stored in the brain, in skeletal muscle, and cardiac muscle. And within those cells, it is stored bound to myosin ATPase. So it is right where you need it in the cell to have this little delivery of ATP to bridge that initial gap. When you start requiring ATP, you can very rapidly keep your ATP supply going through this simple one-step phosphagen reaction. The other pathway that's included in the phosphagen system is our ability to take ADP and kind of move those phosphates around through the enzyme adenylate kinase to make ATP. So you'll notice it is again a single enzyme-mediated reaction for ATP synthesis. All you need is 2-ADP and adenylate kinase, and you can create ATP in the cell. So the phosphagen system is really composed of phosphocreatine and ADP, where we have this very rapid ability to make ATP for a short little period of time, right? We reach maximal yield in about 10 seconds. So we have enough stored ATP for about a couple seconds of an all-out sprint. We have a lot more stored phosphocreatine than ATP, but still we reach kind of maximum benefit of this pathway in about 10 seconds. So this is an important kind of bridge pathway that allows us to keep ATP supply constant, but we're going to need a more high production system to keep ATP going beyond the 10 seconds. So if you put it all together, we have about 10 to 12 seconds. We're going to need some sugar added to this story. So this cartoon of the slide kind of represents glucose metabolism. Really, really important first note under energy of carbohydrates in your notes. Carbohydrates, or glucose, is the only macronutrient whose stored energy can be used to produce ATP without oxygen. I would underline without oxygen or draw some arrows to it. This is really important because oxygen has to get delivered by the blood. And that has to be maintained through the heart and the lungs and the vasculature to get the oxygen there at a constant rate. If we ever have cellular demands that exceed the rate of blood flow delivering oxygen, we need some macronutrient to be available for ATP when we can't get enough oxygen to a cell. And glucose is the only macronutrient whose stored energy can be used to make ATP without oxygen. This becomes extremely important in maximal exercise when ATP need will exceed the rate of aerobic metabolism. So full glucose metabolism has what can be called 2 stages. The first stage takes place out in the cytosol, and that is our anaerobic pathway, the one we just mentioned, that does not require oxygen. It is in the cytosol. These are the enzymes of glycolysis that allows glucose to ultimately get converted into pyruvate, making ATP in the process. As it says in your notes, stage 1, the anaerobic phase of glucose metabolism, accounts for about 10% of the total ATP that you can get out of a glucose molecule. So it's fast, it doesn't require oxygen, but it doesn't nearly get all of the energy out of glucose. In fact, it only gets 10% of the energy out of glucose. Stage 2 is the aerobic phase. It's sometimes referred to as cellular respiration, the use of oxygen in the cell. This is where the pyruvate that came from glycolysis can go into the mitochondria, which is where we use oxygen to make ATP. The processes that go on in the mitochondria are the TCA cycle and the electron transport chain, which ultimately will take that pyruvate and break it all the way down to carbon dioxide and water, releasing the full potential of ATP from that glucose molecule in the process. At this point, let's just take a quick break and just draw this process out, whereby we can get energy out of glucose, or how we metabolize glucose. A glucose molecule is C6H12O6. If we use and have enough oxygen, in fact, we need 6 molecules of oxygen, we can fully break down glucose into 6 water molecules and 6 carbon dioxide molecules. While you do not need to know this chemical reaction as a physical therapist, it can be helpful as you're going through the process of cellular metabolism to have an appreciation of the chemical structure of glucose, what we start with, and what we ultimately end up with as you kind of learn this story. So keep this in mind. I would maybe write that on the bottom of your notes so that you can refer back to just the chemical processes that happen during this pathway. So that represents the complete combustion of glucose down to water and carbon dioxide. Let's first dive in to just the anaerobic phase or glycolysis. So this is the full glycolysis reaction. Let me say right now, in case you find yourself hyperventilating, I do not plan on having you memorize glycolysis. That's not the purpose of where you are in your academic training. I think it's really important to understand the story of glycolysis because it will play a huge role in our lives during exercise. But the names of every nitty-gritty enzyme is less important at this phase if we can take away what this process is able to do, its limitations, and a few other important points along the way. So this entire image of glycolysis is in your notes as a reference. I will go through and individually highlight the specific steps that I want you to know and fully understand. First, if you look at the picture, so we can see the sarcolemma here, so that's the cell membrane. We're in a muscle cell, got some mitochondria floating around, but all these enzymes, which are green on my slide, are in the cytosol. So glycolysis, you don't have to get into an individual organelle to happen. It's right out in the cytosol. So ATP is going to be very near the machinery that will requiring the ATP. It is a 10 enzymatic step reaction. That is only important to keep it in context, especially as we compare pathways. So the phosphagen system was a 1 enzyme reaction. So the phosphagen system is going to be much faster, albeit it burns out really quick. Where glycolysis is a 10 enzymatic step reaction, which is more complex than phosphagen, but not nearly as complex as some of our aerobic pathways. Second point in your notes, right? The entire thing happens in the cytosol. We've hit. The glycolysis has the ability to make ATP very quickly, but it won't last for a super long time. We will talk more about this as we go through, but there are hormones that activate and stimulate glycolysis, as well as the sympathetic nervous system. So as our exercise intensity increases and the amount of sympathetic nervous system increases, you will see a greater activity of the enzymes of glycolysis. So we're looking at the steps that are in your notes here. I'm just going to draw your attention to the very first step of glycolysis. We're going to spend a little time talking about this step because it has some important components. The enzyme that converts glucose to glucose 6-phosphate is hexokinase, and this enzyme plays a very important role in the cell. So you have space in your notes to draw kind of along with me. This might go well in that space just below activated by hormones and CNS, where you have that little blank spot. This is going to be a little bit of a reminder from pathophys or maybe even your human phys class if you talked about it. Here is a cell, and let's make this a different color. In the cytosol, we have GLUT4 transporters. So GLUT4 transporters are not in the cell membrane, so at rest, a skeletal muscle cell is impermeable to glucose. When there is an increase in glucose in the blood, it stimulates the hormone insulin to get released. And on the surface of the cell are insulin receptors. That one got messy. It's a little better. These are supposed to be my insulin receptors. We'll make insulin a perfect little triangle to fit inside of our insulin receptors. When insulin binds to the insulin receptor, there is an intracellular signaling process that tells those GLUT4 transporters, move to the membrane, which is exactly what they do. In response to insulin, we kind of stick these transporters right into the membrane that now makes the membrane permeable to glucose. So I'll make green dots be my glucose in this story. So when plasma glucose goes up, you release insulin. That hormone activates the processes within the cell that move the GLUT4 transporters into the membrane. At this point, why does glucose move into the cell? Do you know the answer to that question? I've got a lot out here, but why does it move? Is it an active transport process? There is a huge concentration gradient, right? Because we've got tons of glucose outside of the cell and almost no glucose inside the cell. So the glucose rushes into the cell because there is this big concentration gradient. Will glucose continue to move into the cell? It would move into the cell as long as the concentration gradient exists, but pretty quickly this is going to equilibrate on either side of the membrane, which would stop glucose from entering the cell. Enter hexokinase, hero of our story. Hexokinase is the first enzyme of glycolysis, and it takes glucose, this glucose molecule. Let's turn back into, so here's glucose, and it changes it a little bit. I'm going to put a box around it to make it look different, and it turns it into G6P. The process by which hexokinase changes glucose just a little bit into G6P does require some energy, requires ATP to do this step. But this is such an important step because as soon as this glucose molecule has changed to a G6P, it is no longer participating in that concentration balancing game that's happening over here with the glucose. So the first step of glycolysis is critical because it maintains the concentration gradient to drive glucose into the cell. If we did not take the glucose and convert it to G6P, it would severely limit the amount of glucose we can move into the cell. So this is the important take-home message of the first step of glycolysis. It is an energy-consuming step. It requires ATP. It utilizes the enzyme hexokinase, and it maintains the concentration gradient of glucose. So I left just space in your notes for this part of the conversation. I'm going to basically kind of redraw glycolysis. You have the detailed version in your notes on the top of the page. The bottom of the page, I'm going to draw it in the way in which it includes only the details that I care for you to know. And I will hopefully try to sell you on why each one of those steps really matters. So we start with a glucose molecule, and we already discussed this first step. That glucose becomes G6P through the action of the enzyme hexokinase and through the consumption of energy to make G6P. G6P then becomes F6P, which ultimately becomes F16P. Now, while the steps between G6P to F16P, that first step doesn't matter as much, the second one is really important. The conversion of F6P to F16P utilizes the enzyme phosphofructokinase. PFK is the enzyme, which stands for phosphofructokinase. Again, please feel free to use these for baby names in the future. It's my greatest regret in life. Phosphofructokinase. Could you imagine how great that would be on the playground? Hey, phosphofructokinase, pass me the ball. Okay, just, 10 years from now, stop in my office and introduce me to phosphofructokinase and you'll make my life. Okay, so why do I mention this enzyme? The only reason, besides it entertains me to say, is that this is the rate-limiting step of glycolysis. So the rate-limiting step is typically the slowest step. It's also the place where we have the control. So when we talk about things that really regulate and control glucose, this is the step that they intervene at. Elevations in ADP in the cell stimulate phosphofructokinase. High levels of ATP inhibit phosphofructokinase, because if you've got tons of ATP, you don't need to be burning through your glycolysis steps. There are many other things. We'll kind of go through things that regulate glycolysis, but just know that this is the rate-limiting step of glycolysis. Okay. From here, something weird happens. So glucose is a 6-carbon ring. Let's just kind of remind ourselves up here. Glucose is a 6-carbon ring. And carbons are ultimately going to become important here, because how many carbons we have left really represents how much energy we still have to get out of the glucose. So we start with a 6-carbon ring, and F16P is still a 6-carbon ring. I promise this does have importance. The one thing I forgot to add in before I move on, this phosphofructokinase step is another step that requires ATP. Let me just jab that in our story before I get too far away from this step. And it reminded me, because this next step that's about to happen is where we actually start making energy through glycolysis. The first half of glycolysis is considered the the energy consuming half. Now we're going to start getting into the energy producing side. Okay, so F16P is the 6-carbon ring, and this next step splits that 6-carbon ring into two 3-carbon sections. Since my words got a little tangled, I will say that a second time. F16P is a 6-carbon ring. This next step is going to split the 6-carbon ring into 2 separate 3-carbon molecules. So F16P will become DHAP, which is a 3-carbon molecule, and GAP, which is a 3-carbon molecule. So we started with 6, and now we have 2 3-carbon molecules. And while it is true in the cell that it becomes these 2 things, it's kind of annoying for students, because as soon as DHAP is formed, it becomes GAP. So you ultimately end up with, let me change my color back, 2 3-carbon molecules of GAP. So we had a 6-carbon ring that we split into 2 3-carbon molecules. So now we actually have 2 GAP. Now, the next series of steps, as far as I am caring what you learn and memorize and know, look like this. ends with pyruvate. If this was biochemistry, there is so much cool stuff in my ... line that we are missing and skipping over because we are physical therapists and we are not biochemists. But for any of you who have a chemistry or biochemistry background, you're probably cringing at this point. To me, the story is much more important, even though I do find it quite cool. Okay, so we started with 2 gap, which means that at the end, we are going to have 2 pyruvate. So for every 1 glucose, you end up with 2 pyruvate because glucose is a 6-carbon ring and pyruvate is 3-carbon. So we don't get rid of any of the carbon in glycolysis, we just kind of move it around a little bit. During the remaining steps of glycolysis, which are represented by a dotted line here, we get ATP out. So ADP gets converted to ATP at one of the steps, and it happens again later again, where you take an ADP and you make an ATP. And since we have 2 of them, for every 1 glucose molecule, you actually would end up with 2 total ATP at this step and 2 total ATP at that step, because technically this gap molecule would go through it twice. The other important thing that happens in this final part of glycolysis, just deciding which color I have not used yet, maybe blue, is we take an NAD and stick a hydrogen on it and make an NADH, well, technically 2 hydrogen. We produce this NADH product, which we're going to go into a lot more detail on in just a minute. But for the purposes of biochemistry, hydrogen is used as an important carrier of electrons because a hydrogen has 1 electron in its ring. So in biochemistry, when you need to move electrons from one place to another, it's handy to move a hydrogen around. So this process by which we added a hydrogen to this NAD molecule was actually removing an electron from the original glucose molecule and sticking it on this carrier to be delivered elsewhere. I'm going to stick a pin in that for right now. We're going to go into a lot more detail in a minute about these electron carrier molecules. But for now, let's look at what we have. We started with a 6-carbon ring. Glucose is a 10 enzymatic step reaction where the first half actually requires ATP molecules, where the second half starts to produce that universal energy donor ATP. The enzyme hexokinase is key for the maintenance of the concentration gradient of glucose, and it is the first step of glycolysis. One thing I would like to add to this picture is we had talked about how we get glucose. Glucose is in our blood, and we maintain plasma glucose levels pretty constant, and we can deliver glucose to the cell through the process of the GLUT4 transporter, and this glycolysis pathway would work very well. But we also have stored glycogen in the cell, and if you had a glycogen molecule stored in the cell, ready for use, and you went through the process of glycogenolysis to rip, kind of a glucose off is what we said, it actually strips off something that actually is G6P. So when glycogen is used as the energy source, we technically start at the step of G6P. That's where glycogen ends up entering the pathway. And this is kind of cool because when we use glycogen for glycolysis, we actually get more ATP out than when we use glucose because we skipped that very first step that required ATP. It's a little bit net neutral because the formation of glycogen required energy, but it's nice right now. A little added bonus. We can have that stored energy molecule that gets a little more ATP out than glucose itself. So whether you start with glucose or glycogen, this 6-carbon ring is manipulated by different enzymes along the way into a new structure that's still 6-carbon ring, which is F6P. The step after F6P is where we split the 6-carbon ring into 2 3-carbon molecules. We get DHAP, which immediately becomes GAP, and a GAP molecule. So for every 1 glucose, we have 2 GAP molecules. So GAP goes through the final stages of glycolysis, which produce ATP and NADH. And since we have 2 of them, we go through it twice. So that means you'd get ultimately 2 ATP from this step, 2 ATP from this step, and 2 NADH when we transfer that electron from the GAP at this stage in glycolysis. This double of the GAP thing always kind of throws students off. So if we redrew, just kind of jumping in at F16P and redrew it where you get a GAP molecule and you get a DHAP molecule, that immediately becomes a GAP. Some of you might benefit from drawing this out as actually 2 separate processes, in case my shorthand confused you. The GAP that came from the DHAP will become a pyruvate, and the GAP that was originally split off of the F16P will become a pyruvate. Let's call this the top GAP. I'm lacking my best way to display this. If I'm in a room with a whiteboard in front of me, I can point to things, which right now I'm feeling a little handicapped. This top GAP molecule, right at one stage, will make an ATP, and at another stage will make an ATP, and will ultimately transfer an electron onto an NAD to make NADH. Similarly, the GAP that was originally stripped off the F16P will go through those same exact steps and make an ATP molecule at 2 separate steps and form 1 NADH electron carrier. So this picture helps make sense of why in the previous drawing I was doing that times 2 times 2. It's because literally there's 2 parallel pathways happening, where when you started at the very top with 1 glucose and you add up this final phase, what you get here is you ultimately get 4 ATP, because this GAP is actually running. There's 2 GAPs going through the exact same enzymatic processes. You get 2 NADH carriers, and you get 2 pyruvate. So if you prefer it written out that way, that might work better for you visually than drawing it as one line where everything is doubled. I hope that helps. This is one that I wish I was in person with you guys, because I know the fact that you end up with 2 3-carbon molecules is a typical area of confusion for students. So if you go back to your notes on the top of the next page, it puts this together. The entire process of glycolysis, when you take 1 glucose molecule, will ultimately make 2 pyruvate. Because the first half of glycolysis uses 2 ATP, and the end of glycolysis makes 4 ATP, the entire process, when you put it together start to finish, yields 2 ATP. So I had that written out in your notes specifically in case that throws you off at all. The reason you net 2 ATP from glycolysis is because you produce 4 and use 2. And then lastly, you yield 2 of these electron carriers, NADH. Now, I want to have a discussion about those carriers, but this video has hit that 50-minute mark, so I'm going to end the video here and pick up the discussion on the significance of this NADH at the start of the next video. Thanks, bye-bye.